For case study 8, week 8, the immunosuppressant drug that I chose was prednisone, and I'm going to discuss the effects it has when entering the body and how it targets the inhibition of T cells and cytokines, more specifically. So prednisone belongs in the corticosteroid class. It's used as an immunosuppressant in renal transplant since 1960s. It works on the immune system to help relieve swelling, redness, and itchiness. So that's used when it's not used as for kidney transplants. It can be worked as a steroid as well. There's been studies and cases where patients have experienced serious side effects when taking the drug, when used after kidney transplants. And because it's a steroid, they experience high blood pressure, high cholesterol, diabetes, bone loss, and increased bone fractures. So there's high risk side effects, but also with kidney and renal transplants, it is used and effective for as a drug to not to suppress the immune system, to not reject the foreign the organ, so the system doesn't see it as foreign. So it works by suppressing the migration of polymorphonuclear leukocytes. It also works by reversing increased capillary permeability. It has an anti-neoplastic effect by inhibiting glucose transport, phosphorylation, or induction of cell death in immature lymphocytes. So as it works as a inhibitor by attaching and entering the cell. It then enters the nucleus and it binds and is activated by the nuclear receptors that are called and referred to intracellular glucocorticoid receptors. The receptors dissociate with the chaperone proteins, and as a result, translocation of the GC corticosteroid and the GR receptor complex moves to the nucleus. It alters the transcription of cytokine genes, and it decreases the activation and proliferation of lymphocytes. So just again, the process which it happens. So after administration, it diffuses across the cell membrane and binds to the cytoplasmic GR, or the receptor. It associates with two HSP90 molecules and FK506 binding molecules to stabilize the hormone form of the receptor, and it inhibits nuclear localization. The receptor undergoes changes and it dissociates from the protein. So it moves the proteins to the nucleus. Once it's inside the nucleus, the complex regulates the gene transcription through signaling pathways. And so that pathway is the IL targeting pathway. So the specifically IL-2 is a T cell derived cytokine that is crucial in the regulation of growth and differentiation of T cells and B cells. Signaling is mediated by the IL-2 receptor that consists of alpha, beta, and gamma subunits. The IL-2R alpha chain increases the affinity of ligand binding, and the IL-2R beta and gamma chains are responsible for the ligand binding and signal transduction. So the IL-2 is, again, a T cell derived cytokine, and it's important for the lymphocyte proliferation and immune response. The IL-2 receptor is a subunit and increases the affinity of ligand binding. Protein tyrosine kinase JK1 and JK3 are associated and are activated after binding the IL-2 to the receptor. Suppression of T cell proliferation and expression of IL-2 inducible genes occur when it's in contact with the glucosteroid mediated inhibition. So how this all connects to the downregulation of immune system. So by inhibiting the transcription of cytokines and proliferation of T cells, this lowers the activity of the immune system. So this is needed so that the immune system does not recognize the kidney as foreign and works to reject it. And then here are my references.